If I ask you to count how many fruits are there, it is very easy because the size of the fruits are large and the number is also small. But if I ask you to count how many pulses are there, then it is very difficult because the size of the pulses are small and the number is huge. The same thing is applicable for microbes also. When someone tries to determine the number of microbes present in a sample, it is very difficult because the size are small and they are too many in numbers. So in order to determine the number of microbes present in a sample, we have to go for a nearly accurate method, but not the same way I have counted the number of fruits. So today's topic is how to determine the number of microbes present in a sample. The samples could be environmental or food or patient sample. In all cases, it is required to calculate the number of microbes present in the samples. Basically, there are two widely used ways by which one can determine the number of microbes. Number one is viable plate count and number two is spectrophotometric or turbidometric method. These two methods are widely used and easy to perform and required less sophisticated instruments. In this technique, the number of viable bacteria per ml in culture is defined as the number of colony-forming units per ml. A colony-forming unit represents a bacterium capable of producing a colony or clumps of cell on the surface of agar medium. Here samples are dissolved in normal saline or in phosphate buffer or sometimes in water to make several dilution in this way. Then diluted samples are plated by two common procedures like pour plate method or spread plate methods. Plates are then incubated at 37 degrees centigrade overnight for growth of the bacterium and to develop visible colonies. And then distinct colonies, or we can say single colonies, are counted. And using this formula, we can easily calculate how many numbers of cells are present per ml. For example, 100 microliter of microbial cell suspension of 10 to the power minus 4 dilution is used for the plating, and after incubation there are 240 colonies. Then what is the number of microbes present? Remember, in this way we can only determine the number of live, or we can say the viable cells, not the dead cells that failed to develop colonies, and the number of cells should be in between 30 to 300. Number of bacterial colonies are 240 by 10 to the power minus 4 into 100, that means 2.4 into 10 to the power 3 colony forming unit per microliter. And this problem appeared in this exam. Now I am going to solve this. Here 100 microliter of samples are plated from 10 to the power minus 4 dilution and number of bacterial cells in CFU are 4. Then what was the stock concentration in cells per ml that was used for dilution? Here I have used previously described equation to count the number of cells present per ml. So 4 by 10 to the power minus 4 into 100, that means 0.4 CFU per ml. In another problem, dilution factor is 10 to the power 4. From this sample, 1 ml is used and 2 plates are prepared. In the first plate, number of colonies are 64 and in the second plate, it is 72. Then calculate the number of bacterium present in the sample. Here first, I have calculated the average number of colonies, that is 68. Now I have used this equation again to calculate the number of bacterial cells present in form of CFU per ml. Here dilution factor is 10 to the power 4, not the dilution, that is 10 to the power minus 4. And volume used is already in ml, that is 1 ml. So colony forming unit per ml is 6.8 into 10 to the power 5. Another widely used method is spectrophotometric method. It indirectly measures the turbidity of the culture. As the number of bacterial cells increased, the turbidity also increased proportionally. And it absorbs more amount of light at 600 nanometer. OD of the sum culture are measured in which no number of bacterial cells are present. Then a standard curve is prepared from these data and it looks like this. In this graph, the OD and the number of microbes are plotted. From this graph, it is easy to measure the number of microbial cells present per ml using this equation or directly from the y-axis intercept. Now, just for an example, suppose the OD value of unknown culture is 0.14. You can calculate the number of cells using this equation. So I have put the value of x, that is 0.14, in this equation and solve the value of y, and it is 5.93. So number of the cells are 5.93 into 10 to the power 7. The third way by which one can calculate the number of microbial cells in a suspension is Petroff Hausser counting chamber. The chamber looks like this when observed under microscope. Several chambers like this. The central square millimeter is ruled into 25 groups of 16 small square. Count all cells within this center square, and the general procedure involves loading cell suspension into a clean chamber and counting the cells within the four corner square or the middle one with the aid of a microscope. Then the count is mathematically converted to the number of cells per ml of suspension. For example, in this picture, there are 10 cells per square. So there are 10 into 25 or 250 cells. In 0.02 cubic millimeter, there are 250 cells, or per 1000 cubic millimeter, the number of cells are 1.25 into 10 to the power 7. Or simply, you can use this equation, number of cells counted into dilution factor into conversion factor. Here, the conversion factor is 5 into 10 to the power 4. So number of cells are 250 into 5 into 10 to the power 4, or 1.25 into 10 to the power 7. And all the methods I have described in this video, if I arrange them according to sensitivity from the highest to lowest, it will look like this. So that's all for now. If you like this video, don't forget to subscribe my channel. You can like, share and comment. And thanks for watching.